Çalışıyor mu? Tamam, çalışıyor. Distinguished session chair, dear participants and friends, I am pleased to welcome you all today. My name is Yelnur Mukhambetzhanov. I am a fourth year student in Arkeology department in English program at Ankara Üniversitesi, and today I will be presenting my topic entitled The Ancient Equus DNA and the Long-Term Adaptation. Böyle mi? Tamam. Before begin, I would like to address the main question of this presentation. How did the Equus genus evolve? What factor influenced this evolution? What genetic markers confirm this? And can we use them to predict the future evolution of these animals? The evolutionary journey started approximately 50 million years ago by forest-dwelling Eohippus, then intermediate forms Orohippus, Mesohippus, Merishippus, and Pliohippus to modern breeds. But the cornerstone question in archaeology till today, it was when the domestication of horses started, and was this a multi-regional process or single regional process? And why archaeologists couldn't answer to this question before? We'll talk about that. So let's start. Classical zooarchaeology methods. The scientists generally measure the remains by osteometry, identifying the species, identifying the pathologies and the surface injuries, and they classify them. But they didn't look into the biological behavior. They couldn't find answer about the historical evolution and adaptation, and they couldn't answer the process of the environmental. So nowadays we have a lot of disciplines which can help us in that moment, and one of them is paleogenetic, which is extracting, sequencing, and analyzing DNA, studying evolutionary history, population, migration, and recently pathogens. In 2021, the Pablo Librado and his team analyzed and combined 273 remains from all around the world, which is shown on the map, from the Siberian part and from the Iberian Peninsula. You can see different colors, which is mean the different clusters of the Equus or horses. Green is a bowtie culture horses, yellow is a modern horses, the dark blue is the Siberian horses or Elan horses, and the purple is the wild European horses. And near to that, you can see the time frame, which is indicate the chronology when these horses lived. And also, this scientist built a paleogenetic tree, which shows the relationship divided into 4 main clusters. And also, they made an affinity matrix, a heat map where the yellow to red indicates the increasing genetic closeness. Here you can see the table with the different clusters, and a higher part is more yellow, which means that the genetic cluster, it isn't similar. And the low part with the yellowish cluster, it's a modern horses. They show that the genetic cluster is the same. Ancestry profiles genetic components for all samples and the key groups like the Przewalski horses and etc. And long time, the Przewalski horses, I mean the ancestors of the Przewalski horses or bowtie horses, thought that there is a first human-managed horses. And the scientists find out that the DOM1 cluster, it is that horses. But in 2021, the scientists find out that the modern horses have different genetic pool from the DOM1 cluster, and it is a DOM2 cluster lineage. And they wanted to find out where they come, the origin of this DOM2 cluster, and why they was so successful, and occupied all territories in Eurasia and not only. To find out the answer for this question, they use the method named locator, predicting of the geographic region where the ancestors of the Domtu or Tarpan and the modern Przeworski horses lived. Here you can see a warmer part which has a different shape with the different colors, and here you can see redder triangular part which is indicate the first and the ancient remains of the modern domesticated horse. And it is located on the low part of the Don River or western part of the Eurasian steppe. Also, you can see here the method called EEMS, or Estimated Effective Migration Surface, which is predicted migration barriers and the average ancestry components found in each archaeological site. Regions inferred as the geographic barriers are shown in shade of brown, and the regions affected by migration are shown in the shade of blue. Before 3000 BC, you can see different clusters, green, dark blue, purple, and less yellow. But between 3000 and 2000 BC, you can see that the other clusters are decreasing, and the yellowish cluster, which is the modern horses cluster, increasing in the Don River bank. And after 2000 BC, you can see that mostly you can see yellowish cluster, and you can see also the bluish part, which is indicate that the modern horses occupied other territories, not by themselves, but also help of the human, because human in the same period they invented the chariots. And let's talk about the extinct breeds. Elan or Equus lanensis, Neon Sax, or the ancestors of the Botai horses or Przeworski horses. Equus giganteus, which is the biggest equid breed in the world, which was. And why we talk about them and acknowledge them? Because they can help us to understand the limitation, biological limitation, and these breeds actually extinct in the climatic changes. And nowadays we can understand why the modern horses was so successful and survived till nowadays. Let's talk about the factors which push the different animals and especially equid to adapt it or to evaluate. It's the climatic changes and the geographical landscapes which played the main role during the million years. But last 1,000 years, the main role played the human touch, because human adapted horses for different purposes and selecting special abilities. Like they're changing their size. You can see that English horses are bigger than the other horses, and the Arabian horses, which is the more smaller, but they are more endurable for hot weather or the areas where there's less water. Let's talk about the main adaptation which helped them, the modern horses, be so successful. There are two genetic genes, which is the GSDMS. It's a gene which has helped the modern horses have the more endurable or withstand back to withstand the riders. And the ZFPM1, which is involved in the mood regulation and the aggression, which help human domesticated them more easier than before. Let's talk about the future adaptations or the adaptations which can see nowadays. Let's talk about the TRPM channels. It's a simpler body molecular sensor, And the TRPM, it's a subfamily which has played the role of the temperature sensor. And the TRPM8 played the role of the cold sensor. And we can see these gens in the mammals and the birds. And for example, the Siberian horses, which had the foal, they survived in the minus 70 below zero. And I suppose this TRPM8 gen helped them to survive in that cold environment. Let's talk about the meaning of the wild. I suppose we should rethink this meaning because nowadays we don't have truly wild horses. And a long time, the Przewalski horses thought that it is the last truly wild horses. But if you talk about their ancestors, they was human managed. And we can say that the Przewalski horses actually rewilded horses. And another example, it is the Mustangs in North America, which come to North America by the colonists and come from the Iberian Peninsula, or simply Spain. To sum up, classical methods are old-fashioned. We need to use the modern technologies to open new ways of development. The paleogenetics showed that the botile horses are not the same as those nowadays. Modern horses are genetically different. The true revolution started by horses with the genetic cluster DOM2. People started inventing chariots and other inventions. The modern horse is the product of intense human selection acting upon specific genetic variation. Humans create a biological partner that reshaped civilization. Here you can see the references which I used during the preparation. And first and foremost, I would like to thank you all for your kind attention. I would like also express my sincere gratitude to International Arkeology Student Symposium for providing me with this opportunity, to Gaziantep University, which is hosting this event, to Ankara University and the Faculty of Language, History and Geography, and to Ankara University Archaeology Society and its representatives for their continuous support. Thank you. Tamam mı?
Çok iyi sunum.
Sesin normal miydi?
Çok güzeldi.